Macromolecules are large molecules that are important to maintain proper function of human physiology. There are four major categories of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. In this lab, we will discuss carbohydrates, lipids, and proteins, which make up the main components of our daily diet. We will use some basic chemical tests to detect the presence of these molecules in solutions. We will be using the following tests for the detection of these molecules: Benedict's test for simple sugars, Lugol's test for starch, uncoated paper test, and Sudan-4 test for lipids. Biuret reagent to test for proteins. For each test, we will use DI water because we know it is pure and does not contain carbohydrates, lipids, or proteins. Because it does not contain any of these macromolecules, it will not react with any of the chemicals used for testing and will thus produce a negative result. Carbohydrates are the most abundant biomolecules on Earth and the chief source of energy in our diet. Carbohydrates are divided into the following three major classes, depending upon their size: monosaccharides, simple sugars consisting of a single sugar molecule; disaccharides, consisting of two monosaccharide units; polysaccharides, chains of multiple monosaccharide units. There are two different tests for carbohydrates: the Benedict's test, used to detect monosaccharides and disaccharides, and the Lugol's test, which is used to detect the presence of polysaccharides. First, we will perform the Benedict's test for simple sugars. Benedict's reagent is an aqueous solution of copper-2 sulfate, sodium carbonate, and sodium citrate. The sugar molecule reduces the copper-2 sulfate, producing a precipitate of copper-1 oxide on heating. The original pale blue color means no reducing sugar. A green precipitate means relatively little sugar, and a brown or red precipitate means progressively more sugar is present. Depending upon the concentration of sugar, we get a range of color in our positive results. For this test, the test solution will be glucose solution, while DI water and starch will be used as negative controls. Obtain 3 test tubes. Add 2 milliliters of glucose solution to one test tube, 2 milliliters of DI water to the second, and 2 milliliters of starch solution to the third. Then add 2 milliliters of bright blue colored Benedict's reagent to each test tube. Place the test tubes in a boiling water bath for 5 minutes. After a few minutes, we may notice a yellow or orange-colored granular precipitate at the base of the test tube with the glucose solution. On the other hand, the tubes containing DI water and starch, polysaccharide, show no yellow or orange precipitate. We can also perform the Benedict's test on an unknown solution. This is the final color we have. Can you tell if simple sugars are present in the unknown solution? The other test for carbohydrates is the Lugol's test, or the iodine solution test for starch. We used glucose solution and DI water as negative controls, and we also tested an unknown solution. First, we add 2 milliliters of starch solution to a test tube using a pipette. Next, we add a few drops of dark amber-colored Lugol's solution. The color of the solution in the test tube immediately changes from dark amber to bluish black or very dark blue. The negative tests with sugar and DI water show no change in color and remains amber when Lugol's solution is added to it. Now, take a look at the result shown by our unknown solution for the Lugol's test. Does it contain starch? Lipids are oily or greasy compounds that are insoluble in water, but soluble in nonpolar compounds such as chloroform. Lipids have a number of uses in the body. Energy storage, insulation, and cell membrane function are a few functions of lipids. Animal fats and plant oils are examples of lipids that we obtain through our diet. A simple test for the presence of lipids in a substance is to determine whether they leave a grease spot on a piece of uncoated paper. Obtain a piece of uncoated paper, such as a brown grocery bag. Draw 2 lines on the piece, dividing it into thirds, and label them water, our negative control, vegetable oil, our test solution, and unknown. On the paper, place a drop of vegetable oil, a drop of distilled water, and a drop of unknown solution on the paper in their respective spots. Set aside to dry for 10 minutes. After 10 minutes, hold the paper up to the light. The water half should appear opaque, while the vegetable oil should appear translucent. What does the unknown solution look like? Another test commonly used to identify the presence of lipids is the Sudan-4 test. It can be used in microscopic preparation as well as to test the presence of lipids in test tubes. With a China marker, number your test tubes 1 through 3. Add 3 milliliters of DI water to each test tube and then 3 milliliters of the following substances: DI water, vegetable oil, and unknown solution. Add 9 drops of Sudan-4 to each test tube. Add another 2 milliliters of DI water to each test tube. What is happening at the interface? Is your unknown positive or negative for lipids? Are these solutions heterogeneous or homogeneous? Many proteins are structural components of hair, nails, muscle, bone, etc. Proteins also include enzymes and transport proteins, which are parts of bodily and cellular function. Proteins are polymers of amino acids linked together in specific combinations. A protein forms when 2 or more amino acids are linked together by peptide bonds, bonding between the amino group of one amino acid and the carboxyl group of another, which form a polypeptide chain. Humans can only synthesize about half of the proteins necessary for function and must consume them in their diet. We will use Bio-Ret reagent to test for proteins. Bio-Ret reagent is designed to indicate the presence of peptide bonds. It will only detect peptide chains consisting of 4 to 6 linked amino acids, and thus will not detect free amino acids. When peptide bonds are present, the solution will turn violet. The greater the number of peptide bonds, the more intense the violet color that persists. Using the China marker, number the test tubes 1 through 3. In test tube 1, we will use DI water, which is our negative control. Test tube 2 will contain egg albumin, and test tube 3 will contain an unknown solution. Pipette 1 milliliter of each solution in its respective test tube. Add 2 milliliters of Bio-Ret reagent to each test tube. Agitate each mixture by shaking the tube side to side. Wait 2 minutes and then take notice of the color of the mixture. You see the positive control, egg albumin, turns a violet color. What color do you see in the unknown solution? Does this indicate a positive or negative result? Macromolecules can be thought of as the building blocks for all living systems. Carbohydrates and lipids are fuel source for our bodies, while proteins are the workers that allow our bodies to break down the molecules and use them as energy sources. Nucleic acids, which we will look at in the genetics lab, provide the blueprint for characteristics and features of all living systems. The combination of all these molecules working together is what builds our bodies and keeps them functioning properly.